Hello, everybody. I'm delighted to be here to talk about insight through analysis and modeling chemical research of the future. I want to start by briefly thanking the organizers who put together a really exciting panel discussion group for these virtual sessions. So I really think that insight through analysis and modeling is the road of the future. As we all know, machine learning and AI in particular have made enormous progress over the last decades, using large data sets to build highly accurate models that have human-like capabilities in fields such as natural language processing and image understanding. And of course, across chemistry, we're increasingly able to assemble large data sets thanks to breakthroughs in high-throughput experimental techniques in particular. This has led to huge growth in the group of researchers interested in applying approaches from machine learning and AI to chemical data across both academia and industry. Of course, there's a number of key challenges. These include how to clean and analyze the data. It's really important to deal with issues such as batch effects and biases in how data is collected. Otherwise, this can have really annoying impacts on the modeling outcomes. We also need to figure out how to represent molecules so that relevant features are available to the model, but the model isn't drowned in enormous numbers of irrelevant features. Choosing the right modeling strategy is key. The model needs to be complex enough to capture relationships in a high-dimensional space, but simple enough to avoid overfitting the training data, which is often, unfortunately, a little bit limited in supply. And it's also important that we build train test splits that really mimic real-world use cases and evaluate accurately how the model will perform in prospective experiments to help us choose which model we should actually test in the lab. So in the remaining few minutes of this talk, I'll discuss some case studies or examples from protein sequence modeling where deep learning has recently really had an enormous impact on the field. So protein sequence modeling is an area that's particularly tractable to approaches from machine learning and AI because of the huge amount of data that's available. This data, of course, is collected from the natural world. It isn't experimentally labeled, but nonetheless, researchers have made huge progress in building models using this data. And I've shown on the right-hand side a plot from EBI, the European Bioinformatics Institute at Hinxton, just outside Cambridge, which shows the seemingly unstoppable accumulation of sequence data over the last several decades. Something like 2.8 billion sequences currently available in the database. So we're all familiar with this sort of canonical viewpoint that proteins are synthesized as a linear chain of amino acids. The problem of going from that linear sequence to 3D protein structure has long been an enormous challenge, and we've seen recent success of approaches such as AlphaFold or RosettaFold that have made enormous progress on this problem. Of course, even once you have the 3D protein structure, we still need to figure out what the protein does. The structure provides an enormous amount of information about the protein function, but it's still a challenge to go from structure to function. Fortunately, nature does countless experiments through mutation selection in order to find good proteins, and we have access to numerous sequences which record this evolutionary trajectory, and a revolution in reading and writing DNA lets us see and understand what nature has evolved. So I'm going to talk briefly about predicting structure from protein sequence alignments. You'll all have seen the headlines over the last few weeks. It's really a very exciting time. I just want to touch on the insights from coevolution that I think really underpin the discoveries that are currently being made. So there's been a really foundational idea in the field of, I guess, biochemistry, whereby compensatory mutations are often observed. So you may have two amino acids that form some kind of interaction in the wild type protein. There may be a loss of function mutant where one of those amino acids changes identity. It's then rescued by a compensatory mutation at a site that's distant in sequence, but close by in structure. And this gave rise many years ago to the idea that perhaps if we looked for correlations across large sequence alignments, we could identify these pairs of amino acids that were close in structure simply by looking at large sets of sequences. So I actually worked on this myself around a decade ago, and essentially the idea is that we compute some kind of score for every pair of columns, and then we look for which pairs of columns, which pairs of residues have high scores, and make the prediction that they will be close in 3D structure. There are numerous measures that you can use for correlation. I'm briefly going to talk about mutual information, which is a local model, and a POTS model, or a global model, which I worked with in some early work around a decade ago, which builds a single model that takes into account or explains all the correlations observed in the data. Now, and the key question is, are the higher scoring pairs as produced by either of these approaches close in 3D protein structure? And what we found was that while the mutual information signal is rather dominated by noise, the global POTS model actually provides a remarkably accurate set of predicted contacts. So this just comes from the sequence data. The predicted contacts are shown in red, and you can see they overlay the crystal structure contacts for this example protein RAS really rather accurately. We're able to fold up the protein given just that information, and recapitulate the structure of RAS and make a number of novel predictions, including the one for the adiponectin receptor that I'm showing on the slide. The structure was solved after we published the prediction, and we were just a few angstroms away over a large number of amino acids. We're also able to make long-range functional interaction predictions for this BAM-A protein, which was then validated. So there was a sort of early signs that even using relatively simple modeling approaches, you'd be able to accurately make predictions about protein structure and function using these approaches. But along came much more sophisticated modeling. DeepMind published this paper back in 2020 after competing in CASP13. They provided numerous features to the model, including profiles, 1D and 2D POTS model parameters, and then basically built a really deep neural network, deep two-dimensional dilated convolutional residual network that contained 220 residual blocks, and really sort of made a step change at CASP13. Lo and behold, at CASP14, they made even more progress. So AlphaFold 2 achieved a median score of 92.4 GDT across all targets. That means that predictions have an average error of about 1.6 angstroms RMSD. It's remarkable. You can see the difference shown in this simple bar plot. The new approach, they sort of went back to the drawing board. This approach learns directly from the sequence alignment, so it doesn't use the POTS model approach anymore. They have a two-track network architecture where they sort of iteratively transform and pass back and forth information at the 1D sequence level and the 2D distance map level. And they use end-to-end learning so they can optimize all network parameters via backprop. It's really a remarkable improvement. This was recently published at the same time, essentially, by the Baker Lab. They extended this to a third track that operates on 3D backbone coordinates and provides a tighter connection between sequence, residue distances and orientations and atomic coordinates. This is yet more progress. This is all happening remarkably quickly. And around the same, just a couple of weeks ago, DeepMind released this huge number of predictive structures for the human proteome, together with 19 other proteomes, something like 365,000 plus unique proteins. It's just a remarkable collaboration between DeepMind and EBI. So talked a lot about structure prediction, but of course, getting to function is really important. And there's a sort of interesting idea that you might be able to go straight from sequence to function and allow, sort of exploit this wealth of data to discover new proteins. Of course, building models for protein sequences is nothing new. We talked about the POTS model, hidden marker models being around for a long time. We actually spent some time training machine learning models to annotate protein sequences. We also used a deep dilated convolutional neural network. Ours had somewhat fewer blocks than the DeepMind one, but worked extremely well. Took in amino acid sequences, unaligned amino acid sequences, and predicted which PFAM class family they belong to. And we were able to show that using both a random split and a cluster split of the data, we could do a better job than the hidden Markov model at accurately classifying held out sequences, both those that are similar to the trained data and those that are distant from the trained data. And our models are shown here in blue. It was a remarkable 10-fold reduction in the error rate made by the hidden Markov model. We also show that the deep learning provides complementary information to existing methods. So you get a bump if you combine the HMM with our network that we call PROTEN. And recently we collaborated with EBI, with PFAM, to add a large number of pre-found matches that were predicted by these deep neural networks. We added about 6.8 million new matches. It's roughly equal to progress over the last decade or so. And we provided the first PFM annotation for 360 human sequences. So that was very exciting. I'm going to talk very briefly about using deep learning to design novel proteins. And this was a project in the context of AAV gene therapy. We're designing capsid proteins. So this large cap gene yields the single cap monomer, which is about 700 amino acids. 60 copies of this monomer form a beautiful, highly structured capsid, which has assemble and package its own genome. So we were able to collaborate with members of the Church Lab at Harvard, who had amazing technology to build large libraries of AAV capsid variant sequences and test them. And we essentially were measuring selections, the ratio of final copies of each variant after a round of viral reproduction and packaging to the plasmid library counts. We figured out that the sequencing counts essentially gave us binary labels, so we could tell whether a variant was viable, like the wild type, or broken. And we wanted to really prospectively test our models. So we wanted to also compare different training sets and model architectures. So we took a cross product between 3 different training sets of very different sizes, 3,000, 9,000, and 65,000 training data points with 3 types of models, and to make 9 different sort of models that we were testing at the end of the day. We used these trained models to screen and evolve candidate sequences. So we ranked 2.1 billion randomly chosen sequences in order to identify a set of really high scoring seeds. And then we followed the model gradients. We walked the sequences. We're using sort of discrete optimization approaches here in order to greedily optimize the models. And we evolved each of them for 20 steps. And we did indeed discover that we could make a large amount of improvement using these models. So the baseline, which is a simple additive model, is shown in gray. And if we used a CNN or an RNN, we found we could do a lot better. Here we're showing how far the sequences are from the starting point on the x-axis and the percent viable or accuracy on the y-axis. You can see that we got a lot of lift with those blue and green curves over that gray additive model baseline. Randomly choosing sequences is shown here in black, and you can see that that really doesn't work at all. This is logistic regression model is shown in orange. For this small training data set, this is for the 3,000 data point training set. The logistic regression model really didn't work well at all, which is a bit surprising because we thought that with limited training data, the simple model would shine. Actually, with the larger data set, I'll show on the next slide, with this middle-sized data set, the logistic regression model did really, really well. It was extremely accurate. It also happens that the sequences it designed weren't very diverse. We did find that neural networks design much more diverse sequences. And so that was a feature that we really wanted in this project. On the other hand, we could have gone back and diversified the logistic regression model predictions. It seems as if here the model really found a ridge in fitness space and sort of walked along it, systematically turning every starting point into its favorite sequence. Or as close as it could get. Then with the largest data set, the logistic regression model also didn't do that well. So it just wasn't as robust of a performer as the neural networks. But we were really excited that we were able to identify sequences that were significantly far from the starting point with all 3 training data sets. This was very surprising to us. We just found that models trained with limited data are able to reliably discover new functional sequences with as many as 50% of the positions changed from wild type. And we found that neural network models were able to discover diverse good sequences, even with limited training data. That limited training data, I should have said on the previous slide, was limited to just single mutants and a random subset of the possible double mutants. So it was also very localized around the starting sequence, and yet we could really extrapolate out from that and walk in sequence space. So thank you for listening. I really look forward to discussing the wide range of work that's going on in protein sequence modeling at the moment. Thanks again.